It's a great pleasure to be here. And so I'm going to tell you a story, perhaps fanciful, but one that I've reflected on in a number of years, which is really how do we die suddenly? I trained as a cardiac electrophysiologist. That's a subspecialty of cardiology that considers how electricity controls the heart, and in particular, at times, makes it go too fast or too slow. And that and one other cause, I really think account for the vast majority of sudden cardiac death. Dr. Cass gave you some statistics this morning, and I think a roundabout number is that about 500,000 people may die from sudden cardiac death, a death from a cause primarily of a defect in the heart every year in this country. So this is a tremendous public health issue, really without tractable solutions, I would argue. So how does one die suddenly? And I'd say that although it's tremendously complicated, and there's lots of people who have studied this, including Dr. Tomaselli and Dr. Cass and others, I think it's possible to break it down into a few simple components, and that's what I'm going to try and do. That the heart could beat too fast to sustain its efficiency as a pump. It could beat too slowly to be a pump efficiently, or it could pop or rupture. So these are a couple real-life examples that I gathered from my practicing life, and this is an electrocardiogram actually worn by a real-life patient who succumbed that was given to me by a friend from medical school who was in family practice outside in a small town in Minnesota. And this particularly unfortunate person came in with a variety of symptoms, feeling bad, and for reasons that aren't entirely clear to me, was given a thing called a Holter monitor to trace his cardiac rhythm. And what this shows is that his rhythm went very badly awry, that he was likely having a myocardial infarction. You can see that because of the changes, the deformation in this kind of elevation of that part of the ECG. And during that period, he also developed what are called extra beats, or premature ventricular contractions. You see them come up sporadically, and then every other one. And finally, it happens in a very vulnerable way, and the heart becomes very fast and irregular, an arrhythmia called ventricular fibrillation. And this is the way that people die suddenly after a myocardial infarction, and this is what happens to the 50% of people with a myocardial infarction that don't survive to get to the hospital. And in fact, it's ability to correct this rhythm with electricity that made cardiac care units actually effective in reducing the rate of death from myocardial infarction. So that's one example of too fast. This is another example of too fast. This is was an unfortunate young man that back in the day when I put in defibrillators, rescuing devices that could block arrhythmias, this is a person with a familial, a genetic form of heart failure, of cardiomyopathy, and I put in this defibrillator, and this is an example of a spontaneous rhythm. Actually, there's some technical reasons why it may not be completely spontaneous, but for purposes of this discussion, he goes from a regular but not healthy rhythm, he also has these extra beats, to a very fast, chaotic rhythm called ventricular tachycardia or ventricular fibrillation. And then his defibrillator makes a decision, it shocks him, and he goes back into this regular rhythm. So those are two examples of sudden cardiac death, this one aborted, the other one not, that happened because your heart goes too fast. But it turns out probably an underappreciated way of dying, especially for people that have very advanced heart failure, is that your heart goes too slowly. And this is an example. This is a tracing, and all you have to know is that this is time moving along like this, and here's these big spiky things are the heart beating, and this is probably slow but okay, but by the time you get to here, there's just not enough of these beats. The heart goes very, very slow, and this patient didn't succumb. It's an example of what can happen when the heart goes really slow. This patient probably would be faint or have even lost consciousness. And then a more rare but also probably underappreciated cause of sudden death from a heart problem is cardiac rupture. So this happens typically in people who have a first heart attack or myocardial infarction, and the muscle tears between the chamber of the heart and its external surface, and blood collects in this sac around the heart. And when this happens, there's really nothing that can be done. People can be taken emergently to surgery. It's heroic. It usually doesn't work. It's a strong marker for mortality. So those are clinical examples of how this might work. And so here I'm going to just consider whether those three seemingly very diverse causes of sudden death could be driven by similar pathways or molecules. And if that were the case, you might be able to develop a parsimonious treatment for reducing the chance of sudden death. And so here I'm going to introduce you to a particular molecule that I've spent a long time studying. I'm going to call it CAMKII 2, and that's because the full name is the multifunctional calcium and calmodulin dependent protein kinase 2, and that's so hard to say. And I would say that just stuff is abundant in heart muscle, and although it's not known why, I think that the best thinking is that it's a kind of turbocharging molecule that allows heart rate to fully accelerate and the muscle function to be fully adaptive. And that's great when you're young and when evolution cares about that sort of thing. But when you get to be my age or even older, it becomes a negative. And this is one of the problems. So this is a cartoon of CAMKII, so we don't really know what color it is. And the top shows here. There's 12 of these in each molecule. A kinase or an enzyme is something that actually lowers the amount of energy it takes to do a particular thing chemically. And this particular thing is to take a phosphate from this energy molecule called ATP and put it in just the right spot on a protein. And CAMKII does that to certain kinds of proteins that broadly control biological responses of heart muscle. What you need to know is that the in-between part, this is the business end, the catalytic domain, and this is the assembly part that allows all these pieces to come together. But in the middle, there's a lot of machinery that regulates whether this enzyme is turned on or turned off. So the biology is very smart, and it's developed a strategy so that the kinase isn't just on all the time. That would be bad, and in fact, that's really what we think contributes to disease. But a number of years ago, we found, sort of like the story that Dr. Cass told, but just in reverse, that there's a pair of amino acids called methionines. They're indicated by this letter M, that those become oxidized. And when that happens, it locks the enzyme into a constitutively active conformation that is linked to disease. And I'm going to tell you a story a little bit about that. So we were interested in CAMKII for a variety of reasons, but it emerged as various people discovered that when they sampled human hearts that had come from people with really, really advanced heart failure, or in animal models of heart failure, or in genetic models, that CAMKII was one of many signals that was increased across all those models and species, including in people. And so this is a series of studies, not from us, but from colleagues and collaborators, Joan Heller-Brown and Don Burrs. And what they did was a very simple thing to say, well, lots of things go up or down in heart failure. You get some sense of that by these complicated diagrams. So here, these investigators said, well, does CAMKII actually contribute to the diseases that we care about clinically, these phenotypes of heart failure and sudden death? And to ask that question, they overexpressed CAMKII, but just in heart muscle cells. And what they found, I think, was remarkable, that these hearts got big and dilated. They didn't squeeze very well. There was a complicated evolution of electrical changes and chemical calcium changes in the heart muscle cell itself. And I think maybe most importantly, where the rubber hits the road, is that in a life chart, this is age of mice, and an old mouse is 2 years. And survival, what you can see is that the more CAMKII that was expressed in heart muscle, the more likely that mouse was to die suddenly. So this CAMKII expression created these twin phenotypes of heart failure and sudden death, very much like the clinical scenario that we worry about. And our group took and this is a long time ago now, took a slightly different tact, which is because there are no clinically available inhibitors for this enzyme at this point, we took a genetic approach to inhibit CAMKII, but just in heart muscle cells, kind of the opposite experiment to what I just showed you. And we linked this up to a protein that glows green so we could identify expression. And these are what the hearts look like. We modeled a peptide after part of that regulatory region, and that peptide is a very selective inhibitor for CAMKII. We made one that's an inhibitor, that's labeled I, AC3I, and we made one that was a control peptide. It didn't have the amino acids right in exactly the right order, and it had no biological activity as a kind of genetic control, and so we labeled that C, control. And we've done many things to these mice, and they have a general phenotype of disease resistance. Here, what I'm showing you is when we gave a myocardial infarction, and Dr. Cass talked about this, but this is when we're surgically, we go in and tie off a really important artery, bringing blood to the heart, and tie it off permanently. So what you can see is this is a genetic mouse with CAMKII inhibition, and it has a huge heart attack because we've tied off, we haven't preconditioned it as you heard about, we just tied it off. And this is a control mouse. They both have huge myocardial infarctions, but what you see is that the control heart is much bigger. In fact, we can't even fit it in this box. And that's because it's dilated, this process of adverse remodeling that we know portends worse clinical outcome and a higher likelihood of sudden death. Whereas the inhibited mouse fits in this box. It doesn't dilate nearly as much. And these are tracings recorded with an echocardiogram, like you saw before, that just show that although the mice before surgery all do about the same, this is fractional shortening here, how well the heart squeezes, that the mice with CAMKII inhibition do much well the 3 weeks after myocardial infarction than the control mice. And although there aren't available drugs, there are experimental compounds, and we don't have to appeal to the FDA to use them in mice. We just used them at a low dose and a higher dose and a control analog, and we found we could create the same benefit by infusing every day a mouse with this CAMKII inhibitor at a higher dose. We could protect the heart from dysfunction after myocardial infarction compared to the other groups. So this seemed like strong evidence that CAMKII contributed to diseases that we care about and perhaps creating a circumstance for sudden death, and that by inhibiting CAMKII, at least selectively in heart muscle, we could produce a clinical benefit that would be an enviable goal and if we could do that in people. So how does that work? And the honest answer is we don't fully understand how it works, but I'm going to tell you some stories that provide some insights into how we think CAMKII inhibition might work. And so this is a slide that really summarizes a series of experiments that we did looking just at that regulatory domain of the molecule and thinking about how clinical therapies that we know work for heart failure might converge on this CAMKII molecule pathway. So we know that the three classes of drugs that really work for heart failure are beta blockers, and here we infused mice with isoproterenol and found that CAMKII inhibition could block some of the heart disease-causing actions of isoproterenol. That works in the beta blocker pathway. The other ones are ACE inhibitors or angiotensin receptor blockers. So that works with angiotensin, and that CAMKII also prevents some of the disease effects on heart muscle from angiotensin. And the third group is a group that works on this so-called aldosterone or mineralocorticoid pathway. You don't have to memorize all those, but the take-home point is that all of those key therapeutics that we know work converge on CAMKII, but they do it by slightly different ways. So isoproterenol activates CAMKII by causing it to stay open through an event called autophosphorylation, and I won't really talk about that much more, so you don't have to memorize that. But I will talk a little bit more about this, that this pathway that we found with oxidation of methionines that lock CAMKII into this constitutively active conformation are activated by aldosterone and angiotensin. So with those thoughts in mind, we thought maybe that's part of how CAM kinase inhibition might be beneficial. It turns out that in heart failure and myocardial infarction, heart muscle cells die, and CAM kinase is one of the executioner molecules that seems to drive heart cell death. And this is an example of an experiment that we published a few years ago showing that CAM kinase inhibition protects heart muscle cells from dying in a paradigm of ischemia reperfusion that you heard about earlier. So that might be one of the ways. And we and others, including Dr. Tomaselli, have mapped out pathways where CAM kinase seems to participate in making heart rhythms go too fast. So there's a lot of work. When I started as a fellow, we focused on this particular protein, a calcium channel. But now we know that CAM kinase appears to regulate all of what we call voltage-gated ion channels in heart muscle cells. And I think that the best evidence for how it contributes to arrhythmia circles around the calcium channel, the sodium channel, and this giant intracellular channel called the ryanodine receptor. And without going into too much detail, what I'd say is CAM kinase appears to act as a pathological feed-forward signal. It's activated by things like disturbed calcium. This is a feature of the heart muscle cell in failure. By enhanced oxidant stress, this is a feature of almost all of the adult cardiovascular diseases we study, and that those things activate CAM kinase to target these ion channels, and that those perpetuate this cycle of activity. And so that's one way that CAM kinase may plausibly activate fast heart rhythms. But what about slow heart rhythms? How could something have an effect on making the heart go fast and slow? And so this is work that was really introduced to my lab by a fellow who's now an assistant professor at LSU, Pari Swaminathan, and he wanted to make a model of a slow heart rate. I actually tried to discourage him because I thought it would be too hard. But he infused mice with angiotensin. This is a hormone that I talked about and that's elevated in heart failure to clamp the mouse's level at about what we see in patients with heart failure. And what you can see is here's the normal heart rate of a mouse that's not angiotensin infused, and you see how slow and irregular the heart rate becomes after angiotensin infusion. And what he discovered is that when you dissected the pacemaker cell area of a heart muscle from angiotensin infusion, that he saw lots of oxidized CAM kinase compared to seeing none in the control group. And when we looked at patients with heart failure and took tissue near the pacemaker region, we found something similar, that patients that had heart failure and had a slow heart rhythm that needed to be corrected by a pacemaker had a lot of oxidized CAM kinase, and there was compared to patients who didn't need a pacemaker and didn't have heart failure, or who had heart failure but didn't need a pacemaker. So it seemed to be a marker. Oxidized CAM kinase seemed to be a marker for a defective heart rate response. And to cut through a lot of detail, we ended up discovering that CAM kinase caused death of these pacemaker cells, and that and this is the pacemaker cell here. This is a picture from a review about Pari Swaminathan's paper, surrounded by regular heart. And normally the pacemaker cells have to inject current to capture the rest of the heart electrically. But when a critical mass of these pacemaker cells die, they prevented the high fidelity capture of the surrounding heart rate, and that led to these really slow rhythms. So with that kind of model in mind, and thinking about another public health problem, which is diabetes, and the fact that if you're diabetic and you have a myocardial infarction, you're about twice as likely to die as somebody who doesn't have diabetes. And we don't really understand why that is, because that difference persists when we factor out the strength of the heartbeat, the degree of large vessel coronary disease, and so forth. And so what Dr. Liu did, who's now a junior faculty member at Iowa, she asked this question: if I make a mouse diabetic and give it a myocardial infarction, what happens? And what she found, first of all, is that this is a non-diabetic, this is percent survival and days after myocardial infarction. And what you can see is a diabetic mouse in orange is much more likely to die quickly than a non-diabetic mouse. And we did lots of studies to figure out why, but the bottom line turned out to be that diabetic mice, and these are mice that have now been implanted with little telemeters so we can measure their heart rate, die with really, really slow heart rates for a mouse and almost no variability. Variability in heart rate turns out to be healthy. Here's a non-diabetic mouse that died from another cause, from cardiac rupture. You can see there's lots of variability. It's a normal, healthy heart rate for a mouse. And these are the expanded scale tracings. So she found that there was more oxidized CAMKII in these mice and also in diabetic patients' hearts. And so we asked a question in a more focused and mechanistic way. Is we wanted to create mice where we could remove these methionines. And so we did that after several years by creating what's called a knock-in model, where we replaced those methionines with valines. Those are amino acids that can't be oxidized. So the CAMKII works otherwise completely normally, but it can't be activated by oxidation. And we found a really stunning result, and that is that these mice with oxidant-resistant CAMKII that were diabetic completely reversed the diabetes attributable risk from CAMKII oxidation, which is shown here in the wild type. So these oxidant-resistant mice looked like they weren't diabetic. And so we found that at least in this mouse model that the extra risk that was conferred after myocardial infarction from diabetes could be reversed just by changing 2 amino acids in this one enzyme. So the final part of the story is heart rupture. So okay, so maybe the heart can go too fast, it can go too slow, but what about rupturing? And this is work done by Julie He, who was an MD Ph.D. student in my lab, who's now a cardiology fellow at Yale. And what she found is that mice that were infused with aldosterone, this is this hormone that is elevated in heart failure and subjected to myocardial infarction, had very high levels of autonomous CAMKII. This is the kind that we think contributes to heart disease compared to other groups. And when she looked at their mortality curves, she found that aldosterone infusion caused excess mortality compared to non-aldosterone infusion. And I have to tell you, just to clarify, although people's aldosterone goes up a lot in heart failure and after myocardial infarction, mice not so much. And so this infusion produced levels of aldosterone that would be seen in patients with heart failure, but not achieved by mice. And we thought maybe the hearts are really, really weak or so forth. But when we did autopsies, we found this kind of yucky stuff here. And when we dissected the hearts, we saw you can see this blanched area. That's the myocardial infarction, and then they ruptured or tore. And this is just a kind of a summary. If you had if you were a mouse infused with aldosterone, there's a high likelihood you'd die from a rupture compared to a wild type. So we repeated that study, but this time with these mice I already showed you that had CAMKII inhibition. And what you can see is that the mice with CAMKII inhibition were much less likely to die than the wild type mice. And when we measured to see, well, how much of that might be due to rupture, we found that CAMKII inhibition only in the heart muscle cells created a difference in rupture frequency from almost half of them to down to wild type levels, despite the continued infusion of aldosterone. So this is really a story that is about CAMKII turning on a gene transcription program to have heart muscle cells produce a matrix modifying enzyme called MMP9 that helps to remodel the outside of heart, but during this vulnerable window of myocardial infarction makes the weakened heart muscle so weak it's prone to this kind of rupture. So just in summary, I'd try and read over there, but I can't see it. So CAMKII, we think the CAMKII pathway is a kind of model for understanding modes of sudden cardiac death, that it appears to be responsible, at least in mice, for diverse causes of sudden cardiac death, and we and others now in pharma think that CAMKII, because of these and other kinds of studies, might be a good candidate for making drugs to try in people. And then I just want to thank This work couldn't be done without lots of people and lots of great collaborators and without support from a number of agencies, most prominently the NIH. So thank you.